Thank you very much, and thanks for inviting me. It's really a pleasure to be here. And before I start my presentation, I didn't want to overwhelm, the presentation, so I didn't include every single data. But after Dr. Horowitz's lecture, I have to mention some co-infections data. As probably you know, back in 2003, 2004, when we started to establish a Lyme disease research group, we had the same question what I heard last night, saying what is the most important question in Lyme disease. And first thing what you do, you read all the articles, and after you sit down and you try to think what is the missing link, what is that nobody is looking at. And I remember back in 2003, 2004, I think that I had two questions. I said, what is the pathogens we're not looking at in ticks, which we might get when we get a tick bite? That was one of the questions. I really felt very strong about it. The other question was why we cannot kill Borrelia. What's going on with Borrelia? This is just not your usual suspect, yes? So we started to work on the first question, absolutely for financial reason, because it's not easy, but it's free to collect ticks. So we went to the woods and we collected, I don't know, probably over 4,000 ticks in the next couple of years. And first we looked at the usual suspect, Borrelia, Bartonella, Ehrlichia, Anaplasma. And we found very high rate for Babesia, very high rate for Bartonella, over 35-40% in ticks. For Borrelia, the first trial was in the high 20s, and after the second year was in the high 40s. And it was a very unusual year. It was in Fairfield County, which is close to New York. We find actually over first 60% in nymphs and over 90% in adult ticks. I never ever able to publish this data. But I couldn't publish even Bartonella Hensel data. You just heard a beautiful lecture this morning, and we found over 40% of the ticks were infected back in 2005 with Bartonella. When I sent it out, they said it's impossible. Hensel cannot be in ticks. So at this point, first of all, lots of frustration, but we were looking for some unusual suspects. It was mycoplasma because of the Horowitz studies, and we find very alarming rate, over 80% for different mycoplasma species. Again, this paper is still sitting on my desk. And the only paper just 2 weeks ago, they were able to publish filarial nematodes in ticks. It's online, so you can get it free. Just Google my name and filarial nematodes. So definitely co-infection is a huge issue, and we keep looking for some unusual pathogens in ticks, just to be sure that when you go to your doctor, you might be able to have a chance at least get tested for it. But the second question, which I'm going to talk about today, is why we cannot kill Borrelia. Let me see, because it looks like it's... Okay. So this is the slide I always start with my lectures, because I want to remind you it is over 30 years when we discovered Borrelia burgdorferi in ticks. Actually, Dr. Burgdorferi discovered it. And today we're still struggling with how to kill it. And again, right after he discovered, we found that a patient has antibodies against Borrelia, and that's how Lyme disease was discovered. And again, we've seen some very good slides about Ötzi. So Lyme disease is here for a long time, over 5,000 years, and I found a very interesting article back in 1600 when John Joselin was walking through those New England woods, and he found that there's some crawling animal who can get into your socks and bite you. So definitely, this is not a problem from 1976. This problem was here long time. Okay. So obviously, the first question when we discovered Borrelia, how can we eliminate it? And there was a lot of in vitro lab studies in the early 1990s, early 2000s. And we discovered a lot of good antibiotics which should work on Borrelia. And as you see, this is just numbers. We call the minimum bactericidal concentration, which means it should kill Borrelia. And as you see, some of the concentration is very, very low, definitely should be achievable in the clinic. But even back in 1989, there was the first studies when it shows that is something wrong. Somehow in a clinic, those antibiotics are now working. There's just few papers from the studies. And of course, we could say, all right, so maybe we shouldn't do in vitro studies, we should do animal studies. And obviously, they started to test some antibiotics in animals. And again, they tried to see some resistance of this bacteria. All right, so how about some stronger antibiotics? How about ceftriaxone, rocephin? It should work. And again, when they did some animal studies, again, some signs of trouble that is even maybe rocephin is not going to work. And that's when people started to look at different forms of Borrelia and maybe hypothesize that maybe different forms has different antibiotic resistance. As you see, this is a oopsie, that was on the bottom. As you see on this right side, that's a famous picture when Borrelia exposed to penicillin, and they start to see some interesting formation forming. So this is now very well known that Borrelia has different forms. This is just a picture we have in a lab as a culture. As you see the spirochete, but always every culture, even with the rich BSK media, has we call the round bodies or cyst forms. So this is not an unusual form. It doesn't need to be Borrelia doesn't need to be stressed to have this form. It's almost like always a backup for the spirochetes. And of course, the L forms, or the McDonald picture, and again, I'm going to talk about the biofilm forms very, very soon. So if you assume that the round bodies has different, maybe antibiotic sensitivity, that of course the first question was, can we see these round bodies in vivo, in the body? And the very first proof was from Dr. McDonald when he seen those round bodies in the hippocampus from Alzheimer's patients. And also some other investigators always proving that it's not just an artifact of the lab. And the Norwegian, as we know, scientist, Dr. Bronson, was spending 10 years to find different antimicrobial agents for those round bodies. The first one was metronidazole, flagyl, and also tigecycline, tinidazole. All those agents look like very effective against those round bodies. Actually, our lab is famous about that. We always repeat every single data. So when we want to repeat Dr. Bronson data, actually we emailed Dr. Bronson, and he was very, very nice, and he helped us to repeat all his data. And it was very interesting when Dr. Bronson just published in PNAS about tigecycline and how effective tigecycline against the round bodies. Just few months later, that was another study, now it's in mice, when they checked tigecycline, and unfortunately they found it's not as effective in vivo than it looked like it's effective in vitro. And of course, we mentioned a couple of times the monkey study from Dr. Embers, 2012, when they also found some persisting Borrelia after treatment, even in monkeys. So obviously the question is, even our best antibiotics cannot eliminate all those different forms. So what can we do? This is back in when we started to ask the same question. Of course, we also looked at the different morphological forms. And the very first experiment, we exposed Borrelia, this is just spirochete, to different antibiotics. And this experiment was penicillin and doxycycline. And as you see, after exposing, yes, we've seen those round bodies as we've seen back in 1989. But as you see also, what we've seen is something on those round bodies, something forming. It looks like some material forming. I have to be honest, at the very beginning, I thought it's just some contamination. You know, you're working with students, you never know what happens, yeah? And I kept asking to repeat it, but we kept saying this. And I remember this is the point when I think I'm emailing daily to Dr. McDonald and ask him to look at those pictures. And he just sent me these pictures from one of the studies, Dr. Eisendal studies, and he said, look at his pictures. This is from infected skin tissues. Look at it, really reminds the one you think is a contamination. This is another picture from this paper, and as you see, it definitely looks like, in that picture you could see the pink one is the Borrelia, the staining is pink, but you see something on it. He called it medusa-like colony of Borrelia. So I was very interested that it's something real, so I was looking every single pictures, which is not just spirochete, in the literature. This is from UConn, University of Connecticut. It's from thick midgut, and as you see, in thick midgut, you see a sort of something forming similar to what we see in the lab tube, but I don't see as much those formations. I'm going to come back to this one a little bit later. Dr. Miklósi pictures, when she stressed Borrelia, she also seen, because at this point, colonies, we didn't really know what is exactly it. So we started to be very interested in these colonies. This is Dr. Ember's paper. I mentioned Dr. Ember's paper. As you see, same here. This is the midgut from ticks, which was feeding on those monkeys. And again, we're seeing something very similar. I call this a photo 51 because this is the one Dr. McDonald sent me back in 2008. And we looked at these pictures, I think, for probably a couple of days before we said something between those two colonies, and it looks like a bridge. Looks like communication, and is one thing which is known about communication and big colonies, big aggregates is called something called biofilm. And that's when we sent out our letter to AJCP, and we suggested that Borrelia might be able to form biofilm. And you're probably going to ask, why this is so important? The most important thing about biofilm in the clinic is it is very, very resistant to antibiotics. It's known to be resistant. Usually, biofilm is a collection of microorganisms. Could be bacteria, could be yeast, could be protozoa, could be single species biofilms, could be multiple species biofilm, could be floating, could be sitting down on some soil surface. The best known of biofilm is your teeth, all those plaques, is practically, this is why you have to go to the dentist every 6 months. I hope I reminded you. And also the shower, if you don't clean it regularly, you start to feel like something very slimy or something very slippery. When I met Dr. Late Costerstone, he told me, he was the father of biofilms, he told me that he believes that over 90% of bacteria can live in biofilm form. Obviously, that still has to be proven, but I think he's right. Biofilm is not an exception. I think biofilm is the rule for bacterial world. So this is just a couple of microorganisms which can find in medical devices. Obviously, a huge problem because that gives you those problems when patient gets these biofilm infections, and unfortunately, especially, we had some problem in our family. One of our dear uncle died after a knee surgery. And this is a couple other common biofilms, which again, very well known and now very well studied. Here's a dental plaque. As you see, is definitely a very complex community. And here's your contact lens, again, if you don't clean it. Think about biofilm is like a city. When pathogens carefully selects locations, actually limits the settlement of too many organisms, divide the labors, store the energy, and I'm going to talk about those exopolysaccharides, which is the slime, and this is very, very good to store energy. of course, transfer some information, lots of communication inside the biofilm, and it is emigration if the biofilm getting too large. So, of course, the question was, how can we prove that Borrelia makes biofilm? Just looking at it is nobody going to believe us, especially, the whole implication, if Borrelia can make biofilm, whether it is really giving high resistance to antimicrobials. So I looked at the literature, and this was the four major criteria that you can call a colony or aggregates a biofilm. The first one is a loss of motility. And, I wasn't sure that's really the best one to start with, because even just a colony without being biofilm can settle down and lose motility. But the big one was the internal rearrangement, which I know it has to be proven. The slime, which is called the extracellular polymeric substance, or EPS, that's the slime, and some communication in the biofilm. So we started to look at Borrelia aggregates, and this is just as early development of biofilm-like structure. As you see, it's a meeting place. Borrelia spirochetes comes together, and they start to form something. And here is if you let this biofilm form, and usually it is the number of spirochetes is the major drive for this biofilm formation. As you see, this colony can be really, really big. And as you see on a very edge of the colony, you could see the spirochetes are forming those aggregates. Inside, as Dr. McDonald mentioned before, you can definitely see some round body forms, probably L forms, and who knows how many other forms you can see here. And this is the 3 stages of biofilm development. As you see, you can see the attachment, the settlement, the growth, and the detachment. And at this point, the one question we ask whether Borrelia really needs to be settled down to make biofilm, and actually that's not true. Borrelia makes very efficient floating biofilms, and that actually was the first thing we studied. And the first picture here, using different microscopy methods showing the early settlement is sort of a mid-phase and a late settlement. On a dark field microscope images, you could see the early settlement. You can see the mid-phase when they're still building it. And the late one is very interesting because you see little islands. And I'm going to come back to these islands because I think it has a very important physiological role. In the DIC, which is differential interphase contrast microscopy, you see the 3 dimension of the structure. And as you see, especially the late one on the second row, you see as a looks like very rigid structure. And it's an interesting thing because if you put this structure between 2 glass slides, it crunches. So it definitely has a hard surface. And we have also fluorescent microscopy pictures that is using a GFP, a green fluorescent protein expressing Borrelia, because I want to be sure that they understand that this is Borrelia, not some other spirochete, and some atomic force microscope image, which I'm going to come back just 1 second. I had one student who wanted to know what kind of matrix requirement needed for Borrelia to settle down, and it shows practically almost everything she studied looks like it's perfect material for Borrelia. So first we saw collagen, one of the best one, but I have to be honest, even Borrelia can settle down in glass. So Borrelia doesn't need really special surface to really settle down. That's what we learned. Let's go back to the atomic force microscopy images. Atomic force microscopy is a very interesting microscopy method. It is using the same magnification as electron microscope with one huge difference. It is what you're seeing is live. For electron microscope, you have to fix the cells, so it's dead. For atomic force microscope, everything you see is live cells. So here's a very first image we found when 2 spirochetes start to get together. The interesting part of this image is you see these flagella-like protrusions coming out, and we still try to understand whether it's an artifact during in vitro culture or something that has a functional role. David Lucky is one of my past graduate students. He loves to play with images. So here is practically a same image, but he used some information from the microscope to show different density of the structure. And what you see here in blue, the white is the spirochete, and the blue you see a different surface forming right on this settlement. And as I mentioned to you, that atomic force microscopy provides live images. So what we're gonna go through next couple images, we're gonna look at the same aggregates making this biofilm. So this is the very first one. As what you see is definitely it is logic to the madness. Yes, it is. You can see how beautifully forming. The spiral is sort of pairing up, and this is one of my favorite images. This looks like a crochet. This is obviously it is planned here. It is not just a random arrangement here. And if you wait, as you see 2 days later, the same colony, you start to lose the symmetry, but you can start to see something else forming. And if you're still waiting, you see that little holes, which Dr. McDonald talked about, that little water channels are forming. And here is just as a late phase of biofilm, you definitely see that those water channels very clearly forming on the surfaces. And this is a composite image. You see that on the side, you still see the spirochete building from outside, and you see that the formation of those water channels, which again for the very known for the biofilm to use for waste and for getting nutrition in. High magnification of the pit, we call it the pit. And the other thing we're seeing is called the protrusion, and I'm going to have a video later. I just don't want to stop it because I hope we don't get it back. But definitely we see not just pits but protrusions. What is the function of this protrusion? We don't know yet, and I didn't see much information in literature about these protrusions for other biofilms. Just another image, and this is the image made the paper when we put together the pits and the protrusion and the whole structure of the biofilm. So it's definitely morphological rearrangement. The next question, can we see those slime forming on the biofilm? And those slime usually it's mucopolysaccharide with some proteins, even some lipids, some extracellular DNA, which we still need to understand exactly the function of this DNA. And those polysaccharide has usually is polyionic, and it definitely can bind to calcium and magnesium, which I'm going to come back to this later. So again, one of our graduate students developed actually used an old technique from 1960s called the Spicer-Meyer technique, which is actually was developed to stain micropolysaccharide, and it is working with 2 different dyes, and give you the acidic, the stain acidic sulfated mucins and some non-sulfated mucins. So that was the image we found. As you see in the middle, we have this huge blue staining, and on the side we have the pink staining. And as I mentioned, this is 2 different dyes. The pink or fuchsin is the sulfated, and the blue is the non-sulfated mucopolysaccharide. So of course the question was, can we identify what is this exactly, especially the blue staining? And for those non-sulfated mucopolysaccharide, the best known is for biofilm is alginate. So the first question was, what we're seeing here with this staining, is it alginate? So we got some alginate antibody, and we stained colonies on the first row. The second row shows a single, this colony. Second row shows spirochetes. This is just a negative control for the reviewer. So as you see, here's a biofilm, and a very strong staining for alginate. This is just a background staining to show you the structure. This is just spirochetes. We just spin it down without any aggregates. As you see, there's no alginate staining in here. This is a negative control when we skipped the antibody for the reviewer. He asked for it. So alginate, what do we not alginate? Alginate binds very strongly with calcium. That's a very known reaction. So the question was, do we have the calcium? Remember that I told you that some late biofilms even crunchy, so definitely suggesting maybe some calcium there. So we had a calcium staining, special calcium staining. And as you see, very strong calcium staining in the middle of the colonies. There's no calcium staining on the edge. And this is some spirochetes here, also no calcium staining. So definitely the middle of the aggregate staining very strongly with calcium. The next one was extracellular DNA, very, very characteristic for biofilms. And now we know that it's not just, of course, you would think DNA, probably some genetic information is coming out from the biofilm, but actually might have even more structural function. If DNA is very, very negative, so one of the ideas that the extracellular DNA actually creates this negative environment and brings in some positive ions like magnesium and calcium. So here's again a specific eDNA staining or extracellular DNA staining. As you see again, the middle of the biofilm staining, the surrounding spirochete are not staining with eDNA. I had also some PCR proof that is definitely some quorum sensing, which is communication is going on in the biofilm. It did not make the paper because the reviewer asked us to take it out. It was just, I think it was just too much information. I'm hoping to finish this study. But obviously the major part, that morphological rearrangement and the biofilm markers are present in the Borrelia biofilm. Of course, the next question is, does Borrelia biofilm has any, gives Borrelia a more resistance to antibiotic? So here is the image which I think that's answers for the question. This culture was treated with doxycycline 25 microgram per ml daily for 3 weeks in the lab tube. As you see, some of the colonies, this is the same field, this is not a composite image, it's the same field. Some of the colonies are dead. This is called the red or live dead staining, which is the red one is the dead cells and the green one is the live cells. So this colony looks definitely very, very dead. But look what happens right next to this colony. It's another colony which very looks like very cystic form, lots of round bodies, and definitely live. So unfortunately, we see this. We call the biofilm heterogeneity. We've seen this all the time, unfortunately. That looks like the antimicrobial, whether it's antibiotic or herbal agent, is working, but we always see some survivors, and it looks like the whole colony either yes or no. Except some colony has some, maybe even inside some heterogeneity. That's definitely something we interested to further study. I'm just going to show you some of the effort to find agent which can eliminate at least some level of biofilms. And this the first one is or studies using doxycycline, looks like tiindazole, tigecycline, amoxicillin, metronidazole. On the slides, and as you see, we look at spirochetes, and as you could imagine that doxycycline is very good for spirochete, as well as tiindazole and tigecycline. Looks like when we combine doxy with metronidazole and tiindazole, we might get even further reduction in spirochetes, and I'll show you data for the round body forms. Similarly, again, tiindazole, tigecycline, as Dr. Bronson suggested, very, very good for those round bodies. Doxycycline is not good for round bodies, as you see, there's a huge increase in round bodies after exposing the colonies, spirochetes to doxycycline. But again, we have agents, as Dr. Bronson mentioned earlier, which are very good for those forms. This is some unpublished information. We checked some other antibiotics, and as you see, depends on the antibiotic. And of course, this is one strain, so you can argue this is maybe other strains might have some different antibiotic sensitivity or resistance. We find that most of the antibiotic has some effect, especially if you combine it with tindazole or metronidazole. You're probably going to eliminate most of the forms. But of course, you want to know, so what happens if we treated a biofilm? And as you see, most of the antibiotic has some effect if you just look at the quantitative numbers. On this one, I think the best effect was doxycycline combined with tindazole, and probably you know that some of the US doctors are trying these combinations, and they reported back that they got some success in some patients. This is another shot trying different antibiotics. Again, as you see, if you do doxycycline with tindazole or metronidazole, you get some good effect. This is after 3 days, so really just a very short period of time. When we looked at under the microscope, again, this is the live dead staining. Red stain is dead, green stain is viable. Tindazole looked practically the best as an individual agent for biofilm. And when we combined it again, looked very promising. Doxy with metronidazole or doxy with tindazole looked very promising for biofilm. So hopefully maybe that's one of the reasons that those clinicians getting some result in a clinic with using these agents. Because tindazole was so effective, actually we tried some other similar class of antibiotics, and they had some effect. Now, probably you're going to ask me, what's the difference between this picture and the previous picture? It is actually one huge difference. This picture is done in a biofilm which was sitting down just on plastic. This picture, we done the same experiment, but it's now sitting down on collagen. We found already that collagen definitely has a protective effect, which I'm going to talk later, that we actually designing some experiment for that. One of my graduate students was very interested in different mutant Borrelia strain to try to understand how doxycycline works, especially how doxycycline might work on biofilms. And this is RPO and RPOS mutant. This is two very famous mutants. This is called the stress mutants. This is not just one pathway. They regulate over 7 different proteins individually and of course together. And as you see, she definitely proven in her experiment that when she using mutant strain doxycycline definitely works better. This is just a Y-type strain. As you see, not much effect with doxycycline. When she worked with RPO and RPOS mutant strain, even at lower dose, she getting better effect. However, Luxus, and it was a very interesting data, Luxus mutant strain is knocking out the communication in the biofilm. So obviously, if Luxus is an important mediator of the communication, taking out this molecule obviously affected very significantly the response to doxycycline. This is one of the papers I'm working on, try to send it out soon. Of course, you can see what other agent you might try. We tried over, I think, 50 different agents. Daily, I'm getting email, try that, between novel antimicrobials, between novel herbal agent or some other agent. I think we tried probably most of it. I want to show you this data. Again, this is showing EDTA effect on Borrelia biofilm. And the interesting data here, on the left side, you see the biofilm is sitting on plastic, just like a dish we use for propagate biofilms. As you see, it looks like high dose of EDTA might have some effect. I think we need to go to 50 millimole, which is quite high. I think that's a 48-hour treatment. That also was very interesting because 48 hours is a long time for a EDTA treatment. Usually, Staphylococcus biofilm, which is studied, gives complete elimination with EDTA within 2 hours. That's, you could see how tough it is, this biofilm, comparing some other biofilms. When we put it on collagen, absolutely zero effect. So collagen had definitely high level of protection. So I wanted to show you more data just very quickly. Of course, we tried different enzymes, different agents. This is just a mixture of enzyme called the biofilm defense, which I think some doctors are using in US. And as you see, has some effect, but it wasn't that impressive that we were hoping for. Of course, you could say, why not just take out the extracellular DNA layer? Yes, we tried with DNase 1 treatment, did not work. Again, you need to know that for Staphylococcus biofilm, it works beautifully. So at this point, we know that we need a Trojan horse, and I'm trying to go fast here because I have 5 minutes. So I know you all feel very guilty about that dessert you just had, and it's interesting that you probably shouldn't, because it's a 2011 paper when it shows that if you combine sugar with some antibiotics, you might get some effect. So we even tried that one. And it was very mixed result. However, one of the sugar we tried is stevia. And I'm going to show you very, very preliminary data. My students still working on this data. Looks like stevia, especially when you combine it with doxycycline, might have very, very good effect. So again, is it a Trojan horse? The Borrelia feels like stevia is food and try to take it in using those channels. We don't know yet. Again, we have a brand new atomic force microscope. We're hoping that maybe visualize this effect using those atomic force microscope pictures. So what's the next step? We still have to prove, however, that this Borrelia biofilm is existing in vivo. And we're using all kinds of different techniques. I got some skin biopsies from Dr. Zalger from Austria. And we have some thick midgut sections from UConn, Dr. Radov. Let me show you just a couple of pictures. This is from the skin biopsies. We found some Borrelia colonies in green, and looks like they're staining with alginate. This is an additional finding, similar sections. The best picture is here. This is again Borrelia, and this is in skin and beautifully staining with alginate. This is just showing the colocalization of Borrelia and alginate. This is another picture, and we have this composite picture we just made not long ago, showing different part of the tissue. We find these colonies all over, and they all stain with alginate. And I know you're going to ask me one more. Oh, we have silver staining proof also, as you see on the right. And or negative control for this kind of experiment is human foreskin. As you see, we couldn't find any Borrelia, or immunostaining has nice background. I want to show you just a few tick midgut pictures. These ticks, they were infected with Borrelia. As you see, we were able to find a Borrelia, but in the right panel, it should be alginate. It's no alginate. So it's no alginate in ticks. It was very the same. This is just a higher magnification. As you see, the colony is right there. Let me just show you. Should be alginate here. There is no alginate. We still have to understand, and we actually proven with even Y-type ticks that we find the colonies, but we did not find the alginate. So it's something about the human body which Borrelia able to make alginate. And of course, you're going to ask me, all right, so other than skin, where else you find Borrelia? And actually, this is a very famous case. This is the autopsy case, and I think that's liver. And on panel E, you see we find a big something which stains with Borrelia, and it also stains with alginate. We just got this week a whole set of tissues from the same patient. So hopefully we can able to analyze multiple tissues, not just skin. And this is Dr. McDonald image using a beacon probe, which he mentioned yesterday. And this is an upcoming paper he's working on. This is a infected liver. This is not biopsy. It is a biopsy. And as you see, we definitely see something which might resemble biofilms. So in summary, Borrelia does form very organized biofilm. It is definitely not a usual biofilm. It shows more resistance to antimicrobials and some other agent than other biofilms. The major component is alginate, calcium alginate, but we believe it's probably other polymers there. Also, eDNA, as I mentioned, very, very important. Definitely has higher resistance than other forms to antimicrobials, and it looks like that we might have some preliminary data that we can find this in infected human tissues. There's a lot going on right now in the lab. Right now, obviously, this is the last sentence is extremely important for us to prove it without a doubt that this is real. This is not just an artifact. Then also, because the experiment on collagen shows that is even further protection against antimicrobials. We have some studies when we study fibroblast and Borrelia interaction. Let's see. We work with Dr. McDonald using novel techniques for those beacon probes because antibodies that's easily to be criticized. So we want to prove some other techniques. I mentioned the AFM studies, and we also looking at other biofilm markers. I have some data on lectin binding studies. Looks like lectin is also a part of it. And the exciting thing is that we were able to work with the University of North Carolina, and looks like we're getting some animal studies going when we can create those biofilm and see what other agent might work in vivo in the mice. And this is my lab. University of New Haven supports us, just built us a beautiful new lab. We were able to get this new atomic force microscope, and I'm really hoping that it really reveals the secret of this biofilm. This is one of the first images we got from using the microscope. And as I mentioned, our research group is we don't have postdocs. We don't have postdocs. We have master level students, so I do a lot of teaching. So it's not just doing experiment, but training those students to be a scientist. And again, the main thing I always tell them that to be very open-minded. Never say never, never say yes or no. Nothing black and white in science. So be very open for interesting discoveries. And this is just my special thanks slides for all those organizations, and of course University of New Haven, who support this kind of studies, and especially for those microscopes, because without that we cannot do our studies. And of course Dr. McDonald, who is never-ending support. Thank you very much.